We are starting Unit 6, which is Immunology. Immunology is the study of our body's defense system. It helps us understand how our body protects itself from diseases like bacteria, viruses, and other harmful organisms. This subject is very important because it explains how vaccines work and how we fight infections. In simple words, immunity means protection. Our body has a system which can recognize harmful things and remove them. Throughout this unit, we will learn about different types of immunity, cells involved, and how the immune system works step by step. This slide mainly talks about how to prepare for exams like GAT-B. It tells us that concept-based learning is very important. Instead of mugging up, we should focus on understanding the topic clearly. Practice questions and previous year questions help in better understanding. Also, regular revision, mock test, and doubt solving are very important. If we study in a structured way, we can score high marks. So always focus on clear concepts, regular practice, and proper strategy. History of Immunology. Immunology started when people observed that once a person gets a disease, they usually do not get it again. This led to the idea of acquired immunity. Edward Jenner did the first vaccination using cowpox to protect against smallpox. This was a very important discovery. Later, scientists like Louis Pasteur and Robert Koch proved that microbes cause diseases. Paul Ehrlich explains how antibodies work. Today, immunology has advanced a lot with modern techniques like monoclonal antibodies and vaccines. Innate immunity is the first defense system of our body. It is non-specific, which means it does not target a specific pathogen. It acts very fast, within minutes or hours. This immunity is present from birth and does not change. It includes physical barriers like skin, chemical barriers like enzymes, and cells like macrophages and neutrophils. These cells kill pathogens by phagocytosis. By innate immunity does not have memory, so it reacts the same way every time. Adaptive immunity is more advanced and specific. It targets specific pathogens and remembers them. Because of memory, the second response is faster and stronger. This is the reason vaccines work. There are two types: humoral immunity, B cells produce antibodies, and cell-mediated immunity, T cells kill infected cells. It also shows features like specificity, diversity, and self versus non-self recognition. Antigens are substances that are recognized by the immune system. They can be proteins, polysaccharides, or any foreign molecules. Not all antigens can trigger a response. Those that are called immunogens. Antigens have small parts called epitopes, which are recognized by immune cells. Some factors like size, complexity, and foreign nature affects immunogenicity. Small molecules called haptens need a carrier protein to become immunogenic. Antibodies are proteins produced by plasma cells. They help in identifying and neutralizing pathogens. Each antibody has a specific structure made up of two heavy chains and two light chains. There are two main parts: Fab region, which binds to antigen, and Fc region, which help in immune response. The variable region is responsible for specificity, while the constant region decides the function. There are five main types of antibodies: IgG, IgM, IgA, IgE, and IgD. Each has a different function. IgG is the most common and can cross the placenta. IgM is the first antibody form. IgA is found in secretions like saliva and milk. IgE is involved in allergic, and IgD helps in B cell activation. Class switching allows antibodies to change function without changing antigen specificity. Molecular basis of antibody diversity. Our body can produce a very large variety of antibodies. This diversity is mainly due to VDJ recombination, where gene segments rearrange randomly. Other processes like junctional diversity and somatic hypermutation increases variation. Class switching also changes antibody function. Together, these processes allow the body to recognize millions of antigens. Antibody synthesis and secretion. B cells get activated when they recognize an antigen and receive help from T cells. After activation, they differentiate into plasma cells and memory B cells. Plasma cells produce large amount of antibodies. These antibodies are mainly in rough ER and processed in Golgi apparatus. Then they are released into blood and lymph to fight infections. In this slide, we study how antigen and antibody bind with each other. The binding is very specific, like a lock and key model. It is also reversible and depends on weak forces like hydrogen bonds, electrostatic forces, and van der Waals forces. Two important terms are affinity and avidity. Affinity means strength of one binding, while avidity means total strength of multiple bindings. These interactions are used in lab tests like ELISA and other diagnostic methods. Complement system is a group of around 30 proteins present in blood. These proteins work in a cascade manner, meaning one activates another in a sequence. It plays an important role in immunity. There are three pathways: classical, alternative, and lectin pathway. The final result is formation of MAC, membrane attack complex, which destroys pathogens. It also helps in inflammation and phagocytosis. Lymphoid organs are the places where immune cells are formed and activated. Primary organs include bone marrow and thymus. Bone marrow produces cells and B cells matured here, while T cells matured in thymus. Secondary organs include lymph nodes, spleen, and malt. These organs help in trapping antigens and activating immune response. They have special regions like germinal centers, where B cells multiply. B cells are responsible for antibody production. They develop in bone marrow and become plasma cells or memory cells after activation. T cells mature in thymus and help in immune response. There are two types of T cells: helper T cell, which is CD4 plus, and cytotoxic T cell, which is CD8 plus. Macrophages are cells that eat pathogens and present antigens. All these cells work together to coordinate immunity. MHC molecules are present on cell surfaces and help in antigen presentation. They are very important for immune recognition and also in organ transplantation compatibility. MHC class 1 is present on all cells and presents internal antigens to CD8 plus T cells. MHC class 2 is present on immune cells and presents external antigens to CD4 plus T cells. T cells only recognize antigen with self-MHC. Antigen processing means breaking down proteins into small peptides. These peptides are then presented on MHC molecules to T cells. This step is very important for immune activation. These are two pathways. Endogenous pathway MHC1 for internal antigens and exogenous pathway MHC2 for external antigens. Cross-presentation is also possible when external antigen is shown on MHC1. Polyclonal antibodies are produced by many B cells and recognize different parts of the same antigen. They are less specific and more variable. Monoclonal antibodies are produced from a single B cell clone and recognize only one epitope. They are made using hybridoma technology and are widely used in diagnostics and treatment. The immune system must be properly controlled to avoid damage. Cytokines are signaling molecules that control immune cell activity like growth and differentiation. Regularity T cells help in suppressing immune response and maintaining balance. If regulation fails, it can lead to diseases like autoimmunity or chronic inflammation. Immune tolerance means the body does not attack its own cell. This is very important to prevent autoimmune diseases. It develops during early stage of immune cell formation. There are two types: central tolerance in thymus and bone marrow and peripheral tolerance outside these organs. If tolerance breaks, it leads to autoimmune diseases. Hypersensitivity means overreaction of the immune system. Instead of protecting, it causes damage to the body. It is classified into four types. Type 1 is immediate allergy. Type 2 is cytotoxic. Type 3 involves immune complexes. And type 4 is delayed response. These reactions are important in understanding allergies and immune disorders. Autoimmunity happens when the immune system starts attacking the body's own cell. Normally, our body can reply to self and non-self, but in this case, the system fails. This leads to damage of tissues and long-term diseases. There are many causes like genetic factors, infections, and molecular mimicry. Examples include diseases like lupus, rheumatoid arthritis, and type 1 diabetes. In these conditions, The body produces autoantibodies or activates T cells against its own tissue. GVHD, graft versus host diseases, occurs when immune cells from a donor attack the tissue of the recipient after transplantation. It is most common in bone marrow transplantation where donor T cells are active. For GVHD to happen, three conditions are needed: donor immune cells, weak recipient immunity, and mismatch in tissue compatibility. It mainly affects skin, liver, and the intestine. It can be prevented by proper matching and immunosuppressive drugs. This slide gives a quick summary of the whole immunology unit. It covers key concepts like innate versus adaptive immunity, antigen antibody structure, and immune cells. It also includes concepts like MHC, complement system, tolerance, hypersensitivity, and diseases like autoimmunity and GVHD. The slide is very useful for last minute revision before exam. Subscribe Brainzz for more latest GAT-B 2026 preparation tips.